Hey, this is Dr. C again, doing this broadcast from my home. This is the backyard, and I thought it's a very nice thing to do. Let's talk a little bit about GMOs. Now, why am I talking about GMOs? We're going to relate a little this eventually to COVID, but God's sakes, I don't want to talk about COVID the entire time. It's all we hear about is COVID. TV, newspapers, it's all it is, COVID. But I'll tell you a little bit on the relationship of GMOs to COVID. Let's start with the basics. What the heck is a gene? What is a gene anyway? Well, it's a functional unit of heredity. This is how you pass it on to your people who come after you. Your genes, your heredity. That's what it is. What is a gene? It's part of DNA. The DNA is on a chromosome. Now, you remember back, well, you probably don't remember back because it's back in 1952 when Watson and Crick identified what the DNA in a human looked like. It was a double curved helix, two DNA helices. Looked like a barber's pole, basically. And on there were a number of genes. And believe it or not, we have over 20,000 genes in our human genome. The genome is all of the genes. I want you to get a little bit of that lingo, you know. You got to have the lingo in order to understand a little bit about it. So that's what a gene is. What else do you need to know? Well, how does the gene produce anything, really? It's a process called transcription. Transcription. How does that work? Well, what your body does, it creates a little RNA. These are smaller molecules, smaller than the DNA, but they can line up along the DNA in the exact number of genes that are on that segment that it lands up with. The RNA, obviously the RNA is smaller, so it lines up with the DNA. Once it lines up with it, it then can take that genome, that little segment of genes, and then through a process of messenger RNA, transfer RNA, and then ribosome RNA, which makes a protein. Now the proteins are what you're made of. This will be your eyes, your eye color, your hair, lack of it, whatever. All of these things are part of what the genes will do. Now obviously there's two genes, one from the male, one from the female. That's how it works. So, with that as a background, what happened now with this gene? How do we evolve to where we are now? It's been pretty much decided so far, that we're not going to use the genes for any reproductive changes in a human. That's pretty much worldwide. They don't want somebody screwing with your genes. Which is probably, again, I'm not saying it's not being done. There are people who are trying to get embryos, people are trying to get stem cells. You know, they're working under the radar, basically, but they're not supposed to. Not supposed to. So, plants have genes too. So, the industry that deals with plants and animals figured, gee, if they could use some of this knowledge about the genes, maybe they could improve what they're doing. And so let's look at a little history of the plants, for example. How do we make our crops, first of all? How do we decide which crops are better? Well, We used to use just the traditional method. You saw that certain plants were bigger, produced better fruit or vegetables. So you picked those, made better seeds with those. And so that was the earliest form, as you might, of the type of gene arrangement. But they went on from that. They went on then to crossbreeding and cross-pollinating. Now usually the cross-pollinating used two different species. And then they would make hybrids. And they would produce seeds from those hybrids. And the plants, again, were bigger and better. So this went on for many years. Remember, it was 1952 that they identified our DNA. It wasn't until 1972 that we began to see some of this gene manipulating after that. Back in roughly about 1976, that's when they began to identify specific genes along our DNA and how they might be able to make a difference in the plant DNA. And so, back then, it was mostly the plant factories that got involved with this. Big plants, Pfizer, Monsanto. Those are the big areas that were involved with the genes. So what did they do? Well, rather than rely on traditional breeding, they tried different things. You can use, for example, radiation. If you expose a plant to radiation, you can alter the genes, the DNA of the plant. That's called mutagenesis. Obviously, not all of the things with mutagenesis worked well. Could go the opposite way. Plant could be worse. Plant could die. Plant could have trouble. But with that mutagenesis, they produced a series of better seeds for their plants. This went on for a while. This was in the 1950s, 1960s. Now they began in the 1980s to do gene modification. Now that they could identify the genes on a particular plant, they could actually isolate a particular gene. Say they isolated a gene that had to do with resistance to a particular insect. Do you know that prior to this time, 20 to 25% of all the plants that were produced for vegetables in this country never made it because of insects and disease? So it's a big loss for farmers. Big loss. Things like that. And at the same time, remember that name Monsanto. Remember that name. They produce a particular pesticide, Roundup. Now, Roundup had a bad name. Not at first. Bestseller. It was a bestseller. But then it had a bad name. Well, what the gene modifiers did is that they took that particular gene in a particular plant and then transferred that gene to the gene in corn and in soy. They made that particular gene that made that plant resistant to disease. It's a natural pesticide. So no longer did Roundup hurt the plant. It didn't bother the plant in the least. And the plant could live despite the insects. Not bad. Not bad. Problem is, initially, what the farmers did then, they said, well, since this isn't going to bother my plant, let's pour the stuff on all the plants. Put it on. Loads of it. Well, this was before glyphosate was then known to be a carcinogen. It caused lymphomas. And they were pouring this stuff onto the plants. Well, obviously this was not the best way to do gene modification, but at least they had then identified that it was glyphosate that did it. And so now they had good resistant plants, and now they were using much less glyphosate, obviously. So that was the genetic modification series. They produced bigger plants, more productive plants, resistant plants. They even had resistance to heat. So if you had drought, for example, those plants would be resistant to drought. Not a bad idea. Not a bad idea. Well, now we come into the 20th century now, 21st century, and roughly about in 2010, we began to see this thing called gene editing. So that's different from gene modification. Gene editing. You'll hear things in the newspaper, and the newspaper reporters don't know squat about it. They're not biologists, for God's sakes. They don't know anything about it, but they write that down. CRISPR, C-R-I-S-P-R. CRISPR. What is that? Well, it's a big name, but what the heck does it mean? Well, what CRISPR means is clustered, regular, interspace, short, palindromic repeats. Well, it's no wonder they call it CRISPR. You can't, obviously you can't remember all those things that it stands for, but it's a CRISPR technique. How does that CRISPR technique work? Well, what they do is they produce this RNA strand. The RNA strand matches the DNA at a particular gene level. That particular RNA strand then has attached to it a thing called Cas9. Could be Cas9, 10, 11, 12, 13, Cas15, could be anything, but an enzyme that's attached to the RNA that will then disable that particular gene. The way the press calls it is it snips the gene out. It doesn't snip the gene out. It's basically it destroys that particular gene. Now you got a little hole in that chromosome. Well, the chromosome can just heal itself. Or what then the biologist can do is to put another gene into that place that will give that plant a particular feature. Maybe it's going to be a little better at something, more resistant to something, whatever. But they replace that gene with another gene. Amazing, the technology that's involved with this. The DNA then heals itself, and then the seed that it produces will now have that in all of the plants that seed produces. Amazing stuff. So now we're in this stage now of gene editing. Gene editing. And so what are the advantages of gene editing? Well, the advantages are you can get a patent on that particular seed. So you may see a seed, a hybrid seed, always demands a higher cost than a regular seed. So they make their money on the seeds. So it's better for business. But in addition, the plant, because of the gene editing and all, has a bigger plant, a better yield of fruit or vegetable. It's more resistant to insects. It has other immunity to plant diseases, may have a better flavor, a better color, all sorts of things that are a result of this gene editing. And so, another big factor. You have a lawn outside. What do you put on that lawn in the spring? You put in a high nitrogen type of fertilizer. And did you know that half of that nitrogen that's in that fertilizer you put down doesn't actually get absorbed by the plant? Half of it. Where does it go? Into the atmosphere, into the soil, eventually into the water. Not a good thing. But the plant only gets half of it. Now, because of gene editing, they can have a bacteria that gets onto the roots of the plant. That would allow the plant then to use that nitrogen to get all of those wonderful things, make it a bigger, greener, more productive plant. Again, a good thing from gene editing. What are the dangers of gene editing? Well, I mentioned right at the beginning, gene editing shouldn't be done for human reproductive things. There's a scientist in China that actually tried to do this, to make this baby that was in the womb resistant to HIV, to AIDS. It was well planned, but didn't work. It didn't work because there are some drawbacks. These aren't perfect gene editings. Takes a lot of scientific knowledge and all to get it to even approach being perfect. I should mention a little bit about B, capital B, little t, BT. That stands for Bacillus thuringiensis. It's a bacteria. Back in the I forget what year it was, but 1976, the BT crops began to come onto the market. It was started back in the University of Washington, I believe. These BT crops, that bacillus contained a bacteria that made a protein toxin. And that helped that particular plant from getting a disease. So a PT plant was good, and a BT crop was even better. The BT crops were mostly corn and soy. And now, roughly 90% of all the soy produced in this country is genetically edited. 88% of the corn, the field corn I'm talking about, not the sweet corn that you eat, the field corn, 88% is genetically edited. Those aren't the only crops that are genetically edited. One of the first ones to go through this process was cotton, and they did it down in the South. They produced bigger, more productive plants. Problem with BT plants, though, that toxin has a structure, chemical structure, very, very frighteningly similar to ricin and anthrax. Now, we haven't seen that in people yet, but it does raise that issue. Are we totally above board with this BT? And do you trust the scientists who are doing it? So, makes you think twice. Makes you think twice. And so, let's go on from there. Who's regulating this? Who in our country is regulating all of these genetic alterations? Well, there's three editors, basically. There's our wonderful FDA. You've heard so much about the FDA. The FDA basically is there to make sure that what is on the market is safe to use. That's what they're there for. Then there is the USDA. They're there to safeguard the safety of plants and animals. Then there's the EPA. Well, how does that get involved? The Environmental Protection, they deal with the pesticides. So if somebody is producing something that is altering the pesticides or doing something with the pesticides, they get involved. So you got 3 government agencies not necessarily working in tandem to try to regulate this whole GMO debacle, basically. So let's go on from there. Let's talk a little bit on the relationship of gene editing now to the coronavirus. The gene editing has a lot to do with the production not only of the tests, but also of the production of vaccine against the coronavirus. And you can envision, I think I told you on the last show that we had, what are vaccines, the types of vaccines that are out there. Some are produced through inactivating the virus. Some are produced by having a live attenuated virus. The live attenuated is better for longevity of the immunity than the inactivated. And as I told you, there are several, there's almost 100 different companies out there trying to produce a vaccine or a test that's immediately available with results. Hundreds of companies trying to do this. So far, there's only roughly about a dozen that have been approved by the FDA. The others, I certainly wouldn't want to have or put my money on the test results. They may be good tests, but they haven't gone through any type of series to test how well they do. And remember, I told you that for a good vaccine or a good test, you have to show not only sensitivity, in other words, how many false positives there may be, people who don't have the disease come out positive, or sensitivity, false negatives, people who have the disease but then test negative. That has to be proven before the FDA is supposed to put that onto the market. Of course, now they all have what they call an emergency use. So, EUA they call it, emergency use, in an emergency. Well, I guess you could call the coronavirus an emergency. We have almost 100,000 deaths now in the United States. Not yet. Another day or two, we'll have it. 100,000. Still going up, but on a lower level in Massachusetts. So, just be aware of that. And I still want to caution all the people in Massachusetts, be aware that the virus is still out there, can still infect you. The sad part of it is that over half of all of the people now in Massachusetts who have the COVID disease are seniors. That's why I'm talking to seniors. That's why you need to still be very careful. The most important thing is social distancing. That's the most important thing. Why 6 feet? If somebody is around, say an orchestra, somebody's singing, that 6 feet is now going to extend to maybe 10, 12 feet. You have to be aware. So wear a mask. Now, when I go out walking in Dean Park, I don't wear a mask unless I'm going to be close to a little crowd. I have the mask in my pocket, I put it on. But boy, I sure like that fresh air. So I keep it off, but I put it on whenever I'm around people where they may get within that 6 to 12 feet distance. Very important. Also important, hand washing. When I go to the grocery, I go to the grocery store. I don't want to change totally my entire life around. I go to the grocery store, but that stuff that I bring back, we clean off the surface, wash your hands, I wear my mask, wear gloves when I go to the store. That's basically what you have to do. All these other nonsense things that people come up with. For example, the people who cut your hair now. Oh, you can't cut the eyebrows. Why can't you cut the eyebrows, for God's sakes? The virus has been found in the tears in your eyes. It had nothing to do with the eyebrows. It goes to show the stupid things that people have to do. Well, they don't have to do it, but the stupid things that the politicians make you do, and they know nothing about the disease. Nothing. So I get aggravated about it, and I apologize for that. But at any rate, I want to get back to the gene editing now in the formation of the tests and the vaccine. Let's talk about the vaccine first. There are several companies now that are trying to produce the vaccine using the Crispr technique. Remember, the Crispr technique produces a little RNA very similar to the DNA segment that they want to do, right on that gene. The coronavirus has several proteins on its envelope. Now, it's not really like skin, it's an envelope. And the envelope has these proteins protruding from it. It's called a spike, SS protein. There are other proteins too that are important on the surface of that virus. One is called the E protein. That stands for envelope. It's just an envelope. It's not like skin. Remember, a virus is useless unless it can get into the body. It has to get into a cell in order to reproduce. If it can't get into a cell, can't reproduce, it ain't going to kill you. So, the scientists, not in China, first of all, the scientists in China have identified all of the proteins, have identified all the genes on this coronavirus. They know them. So what the people who are trying to produce the vaccine are doing, they're trying to get that RNA match up with that segment of protein, with that segment on the chromosome in that plant. And they're using mainly that spike protein, those little spikes, spike protein on that coronavirus. Some of them use 2 or 3 proteins: spike protein, envelope protein, and an N protein. There's also an M protein too, called a membrane protein. But those are the 4 proteins that are used. Why are they important? Because they can act as antigens. Antigens will stimulate an antibody reaction in your body. So, that means that the ones who are producing the vaccine, if they can match those proteins and put that into a vaccine, that hopefully will give you some immunity. Another major way that they're trying to produce this vaccine, the vaccine itself is one thing, but if you can prevent that virus from getting into a cell in the first place, That's another approach that they're taking. How does it get in? The spike protein has different little, I guess you call them foci, little spots on them. One of them will match up with an ACE2 receptor on the cells in our respiratory tract. The further down the respiratory tract you go, the more ACE2 receptors are present. That's why the lungs are the primary target for coronavirus. If you can prevent that S protein from getting into that receptor, blocking it, you've produced a vaccine, basically. You've prevented the infection. So that's another very interesting approach. Very interesting approach. Now, there are other approaches that use this gene editing. Some of them try to block one of those RNAs as they go on to produce a protein. Remember I said you got the RNA, then you have messenger RNA, transfer RNA, ribosome RNA. If you can affect one one of those RNAs, you can prevent the virus from getting in to a cell. All those are interesting ways that all of these different places are trying to produce their vaccines. We should talk for a moment about the testing. What's so good about the test now? Well, it does tell you qualitatively if you have or you don't have. It tells you if you have it. It's very good telling you if you have it. Not so good telling you if you don't have it. That false negative rate, that's been a big bugaboo, especially with that Abbott test, the rapid test. 15 minutes, you got an answer. But we've seen up to 70% false negative rates. Now they try to downplay that. No, we don't have that type of thing. Bull. You don't know just because you're negative that you don't have the disease. If you took that test the next day, who knows, you might be positive. Or if they did it correctly the first time, you might be positive. Or it may be that the reagent that they used isn't 100% effective. All these things enter into whether a test is good or not. So there are companies out there trying to produce a more rapid result from the test, just like Abbott did. I'm sure they'll come up with it eventually. But it is very interesting now as we're talking about getting back into a normal activity again, normal economy again. I don't think we're going to see that this year, a normal economy, but we'll get into it slowly. But as we get into it, the important thing is the testing. Now, they come up with all sorts of excuses why, we only test people who have symptoms. Well, that's fine at the beginning. But you want to know, how large is that population out there? How many of them have the disease? Remember, up to 80% of people don't get symptoms or get mild symptoms. How can you define how many of those people are out there who had the disease? How many? I probably did. I took care of somebody who did. And so, could I get tested? No, I had no symptoms. So, bull. You need to test. Unfortunately, we don't have that number of tests. Unless you have a few bucks, or unless you're a politician, you can get it done anytime you want. The average person like me, I don't have symptoms, you can't get your test. That's ridiculous. In general, we use this term herd immunity. You have to have at least 60 to 70% of the population have antibodies to the virus to get herd immunity in that population. How do we know? Well, without testing, how do you know? Totally ridiculous, isn't it? Isn't it? So I hope that given you a few things that you can think about with some of these interesting GMOs. I'm very interested in the GMOs. I think it's fascinating. I have a Bachelor of Science in Biology. We didn't have this back then. And it's so interesting. I might have even gone into something like that. Instead of being a surgeon, you know, I would have gone into something like that. That's very, very interesting stuff. So, at any rate, remember, 90+% of your soy is all GMO. 88% of the field corn, GMO. A lot of your foods, especially now the plant-based foods that are on the market now, are partly GMO foods. I'm not downplaying GMO. I'm not saying it's bad. But there are advantages and disadvantages to using genetically altered products in general. So that's it for today. I'm very happy to talk to you about this. I hope that I could make it a little easier to understand by giving you a little background in some of the stuff like that. So nice to talk to you, and I'd be happy to have another program again in the near future. Thank you. This is Dr. C.